Hello, how are you today?
Right, how are you?
Doing good, thank you.
So, okay, in this test also there are 35 questions. Wide choice to select your question that you can answer. So that is 25 questions. So the first question is C-value paradox in the eukaryotes. Just a minute. So what is C-value? C-value is the amount of DNA, okay? That is generally measured using an instrument called flow cytometry. Cytometry stands for C, right? So that value comes in C. So since we are all diploids, we'll get 2C value. For example, human genome size is 3.2 billion base pairs, and amoeba is just 670 billion base pairs, like that. Rice is 450 megabases. So the genome size can be estimated using the flow cytometry that gives the value 2C value. So the C value is half of it, okay? So in the bacterial genomes, especially the bacterial genomes are small. So the genome size and the number of genes or biochemical complexity, everything is correlated in the sense bacterial genomes are small. They will have lesser C value, less number of genes, less biochemical complexity. But when we are going into eukaryotes, like plants, animals, the genome size and the biochemical complexity will not correlate, will not be going hand in hand. So since there is no correlation for the genome size, we cannot say, based on the genome size, we cannot say how complex they are. For example, salamanders. Salamanders have 20,000 megabase DNA. But biochemically, they are not much complex. Humans are comparatively much smaller, say 3,500 megabase. So for this question, what we are going to answer is very simple, saying that the C value is nothing but the genome size, first point. The genome size and biochemical complexity in the prokaryotes agrees very well. But the C value is much higher in the eukaryotes and ranges quite a bit. That's why, and then the biochemical complexity in the eukaryotes cannot be explained based on its genome size. That's all you need to write. That is 10 points. Anyone has question? So describe the structure of DNA, including the role of hydrogen bonding and base stacking in double helix. This is a very typical question, right? Very simple question that you guys can answer easily. There is distinct points there in the book. DNA is a double helical and it resembles like a twisted ladder, and the sides of the ladder are composed of alternative sugar, deoxyribose and phosphate groups, phosphodiester bonds between the 3' carbon of one sugar and the 5' carbon of the next are connected through these phosphodiester bonds, and the rungs of the ladder are nitrogenous base pairs, adenine, thymine, cytosine, and guanine. Adenine base pairs with thymine, and it has 2 hydrogen bonds. Guanine base pairs with cytosine, and that has 3 hydrogen bonds. So that is what we need to explain. And then one strand is in 5' to 3' direction, and the anti-parallel strand is 3 to 5' direction. And in the helical structure, double helix makes a complete turn that will have about 10.5 base pairs, and it will have a major groove and minor groove. That is what we need to mention. Anyway, I'm recording all these points so you guys can also listen to the recorded version. Okay, the third question is about the DNA replication. So the leading strand and lagging strand, there are 2 different strands because just now we discussed one strand goes in 5' to 3', and other strand, opposite strand goes to 3' to 5'. So this template differences cause a lot of variation in the sense the one that goes 5' to 3', one that goes 3' to 5' will have a single primer can get added in the beginning. Since this is 3' to 5', the primer also will get attached to it in the opposite direction, that is 5' to 3'. So the primer faces the template, and the template when single RNA primer binds, that primer faces to 3' direction. So DNA polymerase 3 can continuously add nucleotides using the template as information in the sense since this primer is attached to the template, so the new nucleotides can get added easily because 3' end is facing to that. Whereas in the lagging strand, it is different. In the lagging strand, since the lagging strand is in 5' to 3' direction, a single primer cannot be added because in the beginning you cannot add the 5' to 3' strand because the primer will face opposite direction. Okay, so the multiple RNA primers need to be added randomly from the middle. So these RNA primers will have 3' end using which DNA polymerase can synthesize. So we understood the DNA polymerase 3 is like it has what is that structure? Trombone structure. Okay, so this has 2 units, 2 beta units. So one leading strand can pass through normally straight way, but the lagging strand needs to twist around. That's why we call it a Trombone model of synthesis. So, however, in the lagging strand, the synthesized fragments are called Okazaki fragments. So these Okazaki fragments are processed further. RPA primer removes the RNA, and the DNA polymerase 1 replaces RNA primers with DNA. Okay, and the DNA ligase joins all the Okazaki fragments. That's how the leading strand and lagging strand finally will have the same complementary DNA. Okay. So again, the roles of helicase, DNA polymerase, primase, ligase is a different question. Same question. I split it to 2 because DNA replication, writing everything is difficult. So we can write it as 2 questions. So if you prepare for DNA replication and understand, you can answer both 3rd question and 4th question. So what is the difference in the 4th question? In 4th question, you are going to discuss what is the helicase. Helicase unzips the double helical structure. So DNA polymerase 3 is the one that initiates synthesis on the leading and lagging strand in trombone model. And primase is the enzyme complex that adds primers. So on the leading strand, it adds 1 primer. On the lagging strand, it adds different primer. And then once the Okazaki fragments are synthesized, ligase is the one that is going to attach all the Okazaki fragments. So pretty much information is overlapping between these 2. But here what you are going to emphasize is in the leading strand, since it is 3' to 5', this one will have 1 primer. And on the lagging strand, there will be multiple primers because it is in 5' to 3'. And then multiple primers are added randomly at different places by the same chromosome complex. And on the lagging strand, the synthesis happens discontinuously because multiple primers are there. So this, all the primers that synthesize these fragments, I mean, those fragments are called Okazaki fragments. They will be processed. And here you are going to explain various components, DNA polymerase 3 in the beginning, and the DNA polymerase 1 on the lagging strand to repair and match up all the Okazaki fragments, all the patch up Okazaki fragments. Okay. So here, 5 PCR. So outline the steps of the PCR. So what are the steps? Many times you guys did PCR. There is nothing new in it. So the polymerase chain reaction is a powerful technique in molecular biology because that can be used for multiplying any known template, that you have the template DNA for which you have a target sequence you plan to amplify. So the primers are designed flanking to those sequence, and then you will add this DATP, DTTP, DCTP, DGTP, because they are the building blocks for the new template or the new DNA. So tag DNA polymerase is the enzyme that is heat stable enzyme derived from Thermus aquaticus. Thermus aquaticus is the bacteria isolated from the hot springs. So because the enzyme, the DNA polymerase that is isolated from this bacteria that dwells in the hot springs is heat sensitive. So that kind of heat sensitive polymerase is added, and then the buffer is needed because this buffer simulates optimal conditions where magnesium ions, that kind of ions will be there. So the buffer simulates. Okay. Just a minute. Yeah. So the PCR will run in cycles. Okay, first cycle is denaturation, where the temperature is set at 94 to 98 degrees centigrade. So the double-stranded DNA melts into single strands because the hydrogen bonds between the base pairs are broken. And then the next cycle, next step is annealing, where the temperatures of thermal cyclers will ramp down to 50 to 65 degrees centigrade. You see on the denaturing, it is 94 to 98 degrees. Someone put comments here. Are you going to be typing the answer also on the screen? I'm not typing, Victoria. I'm kind of speaking out. So, and also I will send you the recorded version. Okay. So is it possible you could do that? Because she cannot hear me. Can you not hear me yet? Can you all hear me properly?
Yes, I can.
I can hear you too.
Okay.
Yep.
So I will have this recorded version. So typing will take a lot of time. Then we will be sitting here till evening. But if anyone wants, to go through again, I will go through again that question. So in the PCR, it runs in cycles. One is denaturation, where the temperature is 94 to 98 degrees. So the double-stranded DNA melts into single strands. And then the hydrogen bonds between these strands are kind of dissolved or broken down because of high temperature. And then the thermal cycler temperature reduces to 50 to 65 degrees. And then that holds for 30 seconds. So in that 30 seconds time, the primers you synthesized, primers for especially for this PCR from the complementary sequences, these primers will go and anneal at the template. So this, once the primers are annealed to the template, the third step is extension or elongation. So that is 72 degrees typically. So 72 degrees centigrade, that primer, that DNA polymerase will start adding the nucleotides at the 3' end of the primer. Okay, so that is called extension. It's exactly like DNA polymerase, where the primer is added inside our cells whenever the DNA replication occurs. But Taq polymerase adds in the machine, thermal cycler machine, from the priming point, because primers are added at the annealing temperature. So at the extension, it is continuously starts synthesizing. Okay, DNA polymerase starts adding nucleotides at the 3' end of each primer. That's how the new template is synthesized. So now, in the first round, what happens? You have the template that you have added, the DNA amount that you have added in the first time, and then the cycle repeats. Now you have newly synthesized DNA also. So even the newly synthesized DNA forms like a template. This is very important point. So cycle after the cycle, 25 to 35 cycles are set in the thermal cycler. So in each cycle, the DNA gets denatured and primer is added, primer gets annealed, and then the DNA polymerase, that is tag DNA polymerase, will start adding the nucleotides from that priming site. So this one continues 25 to 35 cycles. So what happens? Continuously DNA gets synthesized. And all that new templates also will be templates for the next cycle. In the sense, one strand became two strands. Both these two strands can be template in the next cycle. So the templates are exponentially increasing. So tag DNA polymerase has great role here. That is, it is heat sensitive because temperature every time denaturing temperature is 90 degrees. Normal DNA polymerases will kind of coagulate. The DNA enzymes, DNA polymerase enzyme gets spoiled. So this tag DNA polymerase is very stable, and that can act at temperature 72 degrees. So based on these principles, PCR is kind of designed. So what is the difference between dideoxy Sanger sequencing? Dideoxy Sanger sequencing of the DNA analysis. So this is pretty much PCR. Then what is the difference? So what is the difference between dideoxynucleotides? So dideoxynucleotides are for DDNTPs. What are the DDNTPs? DDNTPs are 3' hydroxyl end is also deoxidized. So what is the difference between ribose and deoxyribose? Because in the deoxyribose, it is 2' hydroxyl end is deoxidized. Whereas ribose sugar will have 3', 2', both hydroxyl groups on the pentose. Whereas dideoxynucleotides, 3' hydroxyl end is gone, is removed. Okay, so this one is called DDNTPs, dideoxynucleotides, 2 times dideoxidized, 2 times deoxy, oxygen is removed. So both of them are H groups. So this deoxyribonucleotides and then dideoxynucleotides, both are mixed in different proportions. So the PCR will continue like we discussed. The template DNA is supplied, a primer is added. So DNA polymerase synthesizes the new strands, but you do not know what kind of DNTP or DDNTP comes into the PCR. So if randomly DNA polymerase incorporates deoxyribonucleic acid or deoxynucleotide triphosphate, like DATP, DTCP, or DCTP, or DGTP, these are normal nucleotides. Then the PCR continues. But by random, because you have mixed in this sequencing dideoxy, that is 2 times deoxidized. So whenever this DDNTP is incorporated, the termination will occur. So that mixture terminates at random positions. Say you are sequencing 200 base pairs, at throughout the length of the 200 base pairs, randomly termination happens. So the termination can happen by the DDATP or DDCTP or DDGTP, anything that is based on the template nucleotide. So different fluorescent dyes are attached to it for all these DDNTPs. For example, if it is DDATP, that fluorescent dye is green color. If DDTTP, that fluorescent dye is red color. DDGTP, that fluorescent color is black. DDCTP, that fluorescent color is blue. So whenever it is terminated, the sequencing machine can sense that what nucleotide is terminated at that position. So using which we can determine the sequence of 200 base pairs. So this sequencing goes very well up to 1000 base pairs. So this is very textbook kind of sequencing, very clean sequence without any errors. So the Sanger sequencing is the first sequencing using which they sequenced the human genome. That is about this method, that is dideoxy sequencing in DNA analysis. So this seventh question is very important question. Outline the steps of gene expression from DNA to protein. So you will have to explain various things here. Steps. So what happens? The genes will have promoter. RNA polymerase is important for transcription. Okay, so the RNA polymerase binds at the promoter, at the Tata box, and then RNA polymerase starts reading the DNA template, the complementary DNA, synthesizing the precursor messenger RNA. And then the precursor messenger RNA is matured by splicing. Okay, spliceosome attaches to the precursor messenger RNA and then starts removing the introns. And a 5' cap is added. The cap is modified guanosine cap at the 5' end that protects messenger RNA. And also it is important for the ribosome binding, okay, in the translation initiation. So 3' polyadenylation is poly A tail is added to the 3' end. That is also for stability and export from the nucleus. So, and the splicing is very important because all the introns, non-coding introns are removed and the coding exons are joined together. And then that mRNA is exported to the cytoplasm where the translation happens. And then in translation also you need to discuss briefly, saying that the ribosome subunit binds at the 5' cap of the messenger RNA and the start codon is recognized, and then the large ribosomal subunit assembles there. That is called initiation. Okay. And then in the elongation, what happens? The elongation factors move ribosome from one codon to the other, and the tRNAs with anticodon enters into the ribosome, and they bring amino acids. And each codon is matched with its anticodon, and the ribosome analysis catalyzes peptide bond formation, or peptide bonds are kind of forged within the ribosome. So the stop codons UAA, UAG, or UGA are encountered toward the end of the open reading frame, and the releasing factors cause ribosomes disassemble. And finally, it releases polypeptide. And then these polypeptides enter into endoplasmic reticulum lumen, okay, because translation is happening on the surface of endoplasmic reticulum. These nascent polypeptides enter into the lumen of the ribosome where the folding occurs. Chaperone proteins assist folding, and then removal of signal sequences, addition of signal sequences will be carried out there, and protein-protein interactions will occur. Various chemical modifications can happen like phosphorylation or glycosylation, and then eventually that will be transported. So this question is divided into briefly talking about transcription and translation, where in the transcription you are talking about various aspects of transcription, that is binding the RNA polymerase to the promoter, and then sequencing, I mean synthesizing the complementary RNA on the sense strand. And then what happens? The RNA, once it is synthesized, how it is transcription is terminated, you can write even that one, like intrinsic termination or row mediated termination. And then once the termination is done, the precursor messenger RNA will get processed, a cap is added, a tail is added, and then splicing happens, removing the non-coding regions. And then that mRNA, which is spliced and processed, is exported to cytoplasm, to the endoplasmic reticulum, where the translation happens. So in translation, initiation is 5 small ribosomal subunit with the initiator tRNA, which is methionine, will get added. And this complex is recognized, will scan through and identify AUG, start codon, where the larger subunit also assembles. tRNA brings amino acids to the ribosome, and then each codon is matched. Each codon is matched with tRNA's anticodon. So this catalyzes formation of peptide bond between the amino acids. And during the termination, UAA, UAG, UGA are reached. And release factors cause ribosome to release, and post-translational modifications are protein folding happens, chemical modifications occur, and then the transport to the functional location can happen. Chaperone proteins will help, of course, folding those proteins. So that is what you guys need to discuss for the outline of gene expression from DNA to protein. Okay. So for the 8th question, the role of RNA polymerase 1, 2, 3. This is very simple question and easy to answer. Because RNA polymerase 2 is protein coding genes. All the protein coding genes, RNA polymerase 2 is important. RNA polymerase 1 is important for all the ribosomal RNA, especially 28S, 18S, 5' RNAs. Okay, and that ribosomal RNA is localized in the nucleolus, where in the nucleolus it gets added to the various nuclear proteins and assemble smaller and larger ribosomes. So in short, RNA polymerase 1, it synthesizes 28S, 18S, 5.8S. And then you can localize these RNAs in the nucleolus where the ribosomes are assembled. Whereas RNA polymerase 2, as we discussed, that translates messenger RNAs. That transcribes or synthesizes messenger RNAs, precursor messenger RNAs, and also small nuclear RNAs. Okay. So the small nuclear RNAs and microRNAs regulate translation. You remember we discussed that. So RNA polymerase 2 is responsible for synthesizing the messenger RNAs and also small nuclear and microRNAs. Okay. And then what is the role of RNA polymerase 3? All the tRNAs, transfer RNAs that we just discussed in translation. So the RNA polymerase 3 synthesizes this tRNAs, 5S RNAs, and small non-coding RNAs as well. So these 3 different eukaryotic RNA polymerases are very important. So that's the 10 points question. Very easy to get points there. So describe RNA processing units. Again, it is same question like question 7. A part of question 7 comes here. So if you prepare for one topic, you can answer 2-3 questions. That is the trick here. So what happens when the precursor messenger RNA is synthesized? A cap is added. That cap is called 7-methyl-guanosine cap or M7G cap. So that is added at the 5' end. What is the purpose of this cap? This cap protects messenger RNA from the degradation by exonucleases. Exonucleases, if allowed, they degrade messenger RNA from 5' end. So if there is a cap there, exonucleases cannot attack that messenger RNA. Okay. So the ribosome recognition and binding also very important. This cap also helps in translation where the ribosome binds there. And also for the transport proteins can bind and help that messenger RNA to reach endoplasmic reticulum. That is the 5' capping. And then splicing is very important. What happens during splicing? Non-coding introns are removed. And what are the coding parts? Exons. So they join together. So a large complex of spliceosome is there. U1, U2, U4, U6. So these are all very important because they can assemble intron-exon boundaries and form the lariat. And at the lariat point, this spliceosome like U2, U4 can join and then cause transesterification. Transesterification removes the introns and then exons are joined together. Also at this point, alternative splicing can happen. So combinations of exons produce multiple transcripts, a variety of transcripts that eventually translate into multiple proteins. So that is all under spliceosome. So what happens at the polyadenylation? So a stretch of adenine nucleotides, that is poly A tail, is added. About 200 poly A's will be added. Adenine nucleotides are added to the 3' end of the transcript. So what is the use of it? It enhances stability and also it aids nuclear transport. So some proteins motor proteins can bind, some of them can bind at the 5' cap or some of them bind at the 3' cap and they can drag to the cytoplasm where translation will happen. And also for translation initiation also it is important. Or translation termination, also poly A tails are important. So that is about 3 aspects. Okay, capping, splicing, and polyadenylation. So question number 10 is mechanism of translation initiation. Now you need to discuss translation little bit more detail in this question. Because if you are prepared for transcription and translation, this translation initiation also can be studied. So what is most important in the translation initiation? There is this small ribosomal unit, 40S. It binds to various initiation factors. These initiation factors and the smaller ribosome unit forms a pre-initiation complex along with the tRNA methionine. Always tRNA that is attached with methionine alone can be initiator tRNA. Okay. So when all these initiation factors like EF1, initiation factor 4, initiation factor B, initiation factor A. All this will attach to the smaller ribosomal 40S unit, okay, along with the methionine specific tRNA. It becomes like a 48S initiation complex. So this one can bind to the cap, 5' cap of the messenger RNA. So once this binds at the cap of the messenger RNA, this complex can scan through to find the AUG start codon. So what is the difference between the eukaryotes and prokaryotes? 5' UTR, that won't get translated. First exon is 5' UTR. That will have a unique sequence for each gene. Whereas for bacteria, it is Shane-Delagarno sequence. So this is initiation, okay? And then larger subunit that has A site, P site, and E site will get assembled. So this is called translation initiation. So you have in the book beautiful diagrams, and also in the PowerPoint, you have things. So you can kind of cross-verify and listen to my notes again, what I am discussing here, and come up with your own synapses. I'm just giving you guidance here, what should be there in the answer. So here is transcription regulation, question number 11. That is negative and positive transcription regulation. So in prokaryotes, what happens? Sorry, this is question is regulation in prokaryotes and eukaryotes, because negative regulation happens in the prokaryotes. So what happens there? The genes are continuously on, but repressed with what? A repressor. So there are two kinds of regulation there. Inducible repression and repressible repression. The lactose is inducible repression, where the lactose is inducer. It binds to the repressor. And then, whereas repressible regulation occurs in the tryptophan, where tryptophan and the aporepressor makes complete repressor. Once the tryptophan is totally expended, that falls off automatically. So both of them are negative. Whereas in eukaryotes, it is always positive regulation. So what happens in the eukaryotes? There has to, mostly the default state of the genes are off. So first point you need to mention for this question 11 is default state of prokaryotic genes is on, and default state of eukaryotic genes is off, okay? So how this switch off genes will get switched on? That is appropriate activation proteins needs to bind. For each gene, there will be upstream or downstream activation sites. So these activation proteins need to bind to activate it. And here is the question number 12, is the lac operon. So how the lac operon responds to the presence or absence of lactose. So what is the lac operon? Lac genes, beta-galactosidase, transacetylase, or permease, are three different lac genes are under the control of this operon. Operon will have promoter and operator, and also LAC-I. LAC-I synthesizes repressor protein, and then that repressor binds at the operator. So that's how it is switched off. So that switched off mechanism occurs when there is absence of lactose. So when there is absence of lactose, there is no need to lac operon to function. But when there is presence of lactose, this lactose can bind to the repressor and can make sure that the repressor is kind of disassociated, or it induces the gene to function. then what happens? The lac operon functions producing all three enzymes that beta-galactosidase can break down this lactose into glucose and galactose. Okay, and permease can bring more and more lactose inside the cell. And transacetylase is important for further utilization of glucose and galactose. But what happens initially, how this system is switched on when there is no lactose inside? When absolutely there is no lactose inside, but how this lactose lac operon system is maintained through a protein called CRP. What is CRP? CAMP receptor protein. So all the ATP is converted to adenosine monophosphates. When it is starvation happens, cyclic AMP gets accumulated. So there is a protein called cyclic AMP receptor protein that can bind to the repressor and can jumpstart this lac operon. Okay, start bringing lactose inside the, because permease is produced that can bring some lactose inside the cell, and this eventually the lactose can form like a strong inducer. It can bind to the repressor very strongly and can jumpstart regular mechanism. So the lac operon system has a bypass mechanism, two mechanisms. One is lactose can induce it, but in absence of lactose, CAMP receptor protein can induce it. Folks, are you following what I'm talking? Is it helping? Please post in chat windows. Is it helping you? Of course, you need to listen to this recorded version. So Kaylee and Brandy, Natasha. Please type, all of you, what, oh, it helps for Terence too. Victoria, are you still there? Looks like some of them left. That's okay, I will send the recorded version again. So what is the mechanism of attenuation? That is question number 13. So what is attenuation? After operator, there is a sequence called leader in the trip operon. That leader is 114 base pairs or 114 nucleotide length leader. Once that is transcribed, it will produce a stem loop structure. The stem loop structure will have 4 domains in that 114 nucleotide messenger RNA can get folded and produce like loop and stem loop structure. 1 and 2 are stems and 3 and 4 are stems binding to each other. In that orientation, transcription is off. So what is important is there should be translation of this loop. So the ribosome needs to pass through this 1 and 2, and then that opens up 1 and 2 stem, creating a kind of modification of stem loop structure between 2 and 3. So formation of 2 and 3 stem indicates that stem 1 is released, domain 1 is released, and domain 4 is released. That starts transcription. Okay, so there has to be, there are 2 tryptophan codons in that attenuation process in that leader messenger RNA. Those tryptophan codons needs to be passed by the ribosome. So when the ribosome is entering through those stem loop structures, there need to be tRNA tryptophan that moves that ribosome further, creating kind of anti-termination loop. So the leader will form a termination loop that is 1 and 2 and 3 and 4 binding to each other. So when the ribosome passes through this loop, there will be anti-termination loop that is between 3 and 4, 2 and 3, releasing 4. That release of fourth domain of this leader is very important for transcription. So this process is a fine control or fine tuning of tryptophan operon. So this process is called attenuation. Okay. So what is the roles? Question number 14. What are the roles of enhancers and silencers? So there are binding sites for proteins, very short proteins, almost like 20 nucleotide, 20 to 30 nucleotide length binding sites for enhancers or silencers. So some proteins will bind there, enhancers proteins or silencers proteins will bind there. So the DNA will loops around, bringing those enhancer or silencer specific proteins closer to the transcription complex. So that looping around and then physical kind of activation will happen between the enhancer proteins with the transcription complex, or silencer proteins with the transcription complex. Enhancers enhance the transcription, silencers reduce the transcription or silences the transcription. So how far they can be? They can be very far away from the promoter, maybe sometimes 1 or 2 kb away, but the DNA bends around, bringing that enhancer protein closer to RNA polymerase complex. Okay. So what is epigenetic regulation and how DNA methylation influences gene expression? So what is the epigenetic regulation? This one is a very important question. That histones that are in the nucleosome are kind of methylated. And what happens once that they are methylated or deacetylated? So the histones will have tails. Those tails will have amino acids, especially lysines and arginines. They can undergo acetylation or methylation. Okay. So what is the impact of that acetylation or methylation? Acetylation is the mark of transcription activation, whereas methylation is that methyl groups, CH3 groups added to the lysines, they can silence the genes. So there are enzymes, DNA methyl transferases or histone methyl transferases. They can add methyl groups to the DNA. In the DNA, they add to the cytosine. If it is histones, they will add to the lysine or arginines. So the gene silencing occurs, and this silencing kind of represses the transcription. There are acetylation also can happen. There are special enzymes, kind of acetyl transferases, and they can remove these methyl groups, and then they can acetylate these lysines and arginines. This acetylation activates the gene. One represses, one activates. So this occurs at the chromatin level. So this is called, this is very important for the epigenetic regulation. And then the epigenetic regulation, especially at the histone level and the DNA level, they communicate. If the histones are getting methylated, DNA also will get methylated. That is, cytosines will get methyl groups. And then the gene is kind of silenced. So that is 15 questions so far. And what is RNA interference and its role in the gene expression regulation? So we understood that the chalcone synthase is the gene that was first identified as for RNA interference, that is petunia flowers. They are purple in color. So what is the purpleness due to? That is because of anthocyanin pigments. So these anthocyanin pigments are made from the chalcone synthase, a gene called chalcone synthase. So somebody by mistake flipped the chalcone synthase in opposite direction, that is 3' to 5'. That produced a double-strand RNA. So that double-strand RNA can get diced and it can interfere in the translation. Later, after this discovery, they identified microRNAs. MicroRNAs are the genes. These microRNAs, what can they do? These microRNAs also will form a double-strand RNAs, be looping around stem loop structures. These microRNAs are diced by dicer enzymes. 21 nucleotide fragments, they get loaded to the RISC, RNA-induced gene silencing, RNA-induced silencing complex, RISC, RNA-induced silencing complex. So this will have a protein called argonaute protein, and this argonaute protein, or RISC, can deliver this passenger strand, which is bottom strand of the double-stranded fragments. That bottom strand can get loaded to the translational machinery, where the messenger RNAs are getting translated. In other words, passenger strands of microRNAs can bind to the messenger RNAs that are about to get translated, and then that binding kind of stops translation, or translation is regulated, or translation is impaired. So this is interference. Okay. So distinguish between the genomics and proteomics and how they complement each other. So this is the question coming from question number chapter 10. What is genomics? Genomics is a whole lot of DNA sequence. Especially the genomes can be sequenced using next-gen sequencing, like sequencing by synthesis, or circular DNAs that in the PacBio, many nanopore sequences that uses protein motors. So a lot of DNA sequence is generated, and how that can be processed, that has to be processed through bioinformaticians or comparative genomics. So once you sequence a new DNA, that needs to be understood. In other words, that process is called annotation. How that is annotated? By comparative genomics. Like you are sequencing a new organism, a plant, a new plant, and how do you understand what is there in that genome sequence? That can be compared with already previously existing annotated genomes, like Arabidopsis or rice, and can be understood what kind of genes are there in the structure of the genome. So for the genomics, most important thing is comparative genomics. You need to compare those DNA sequences, comparative analysis, annotation. And what is proteomics? So how that is different? So the genomes especially identifies number of genes, the gene structure. But using that, you cannot annotate proteins, because a lot of alternative splicing happens. Once the transcription occurs, that precursor messenger RNA gets processed into fragments, like a set of exons will form an open reading frame, and they produce unique proteins, and then the proteins and proteins interact. Lot of protein interaction happens. So most of the enzymes and metabolites are hollow enzymes. In other words, they have a lot of subunits. Each subunit is coming from one protein. Okay. So the proteomics how can we develop the data? LCMS, this kind of instruments, they can sequence proteins. So that is called peptide sequencing. So the genomics is coming from DNA sequencing, and proteomics is coming from peptide sequencing. Sometimes RNA sequencing, that is sequencing just transcripts, will help us to predict their proteins to some extent, to a large extent, in fact. Lots and lots of RNA-seq or expressed transcripts can be used to predict those proteins. So not only peptide sequencing, but also transcriptome sequencing will help to generate a set of proteins in a particular situation, like cancer cell and then healthy tissue. When you compare cancer tissue and healthy tissue, you can do messenger RNA sequencing and can predict what kind of proteins are there in cancer tissue comparatively to the healthy tissue. So finally, they complement each other. So once you understand the proteins that are coming from, and then you can clearly link it with the genomics. So both genomics and proteomics are important for one final goal that is called annotation. Genomes can be annotated. Okay. So here is question number 18. So describe the basic principle of CRISPR-Cas9 and the genome editing. So last class we discussed what is CRISPR. So CRISPR is a part of bacterial immune system. What is definition of CRISPR? Clustered, regularly interspaced, short palindromic repeats. So a Cas9 is a motif that is attached to the CRISPR that is called CRISPR-associated protein. So Cas9 is just one kind of that. So Cas9 is attached to the CRISPR. So what happens there has to be a guide RNA. So this guide RNA is designed from the gene of your interest, and then it is attached to the it is attached to the tracer. Okay. So what do we have in the CRISPR? CRISPR will have repeats. So what do we have within the repeats? Spacers. So what are the spacers coming from? From various viral DNA, right? Viral DNA, once it is chopped, it is integrated into the CRISPR. And where is this CRISPR in the bacterial immune system? That is attached to Cas9. The most important part of CRISPR is what? Tracer or trans CRISPR motif. So tracer sequence need to be added to the guide, like you want to edit a gene. If you want to silence a gene or genome editing, you need to design a guide RNA from the genome sequence to the genome sequence of the gene that you want to silence. That gene-specific sequence, a short RNA sequence, need to be designed. That should complement to the DNA target. Okay, so that is attached to the Cas9 gene, and then a tracer. So what is the use of tracer? Tracer is the only one that attracts Cas9. So you just need to have guide RNA and the tracer attached to it. And the Cas9 is, of course, separately put in the construct. So Cas9 is translated in the cytoplasm and brings back to the nucleus, whereas the short RNAs are kind of transcribed within the nucleus. The Cas9 attaches to the guide RNA because guide RNA has tracer. Okay, so this whole complex guide RNA and Cas9 can attach to the DNA sequence, the target DNA sequence. Especially one thing we did not discuss in the class is PAM. So what is PAM is a 3 nucleotides that is very specific to Cas9. Each Cas, it could be Cas1, Cas2, Cas3. So many different Cas are there, like CRISPR-associated proteins that can create double-strand breaks. So for each Cas, there is a signature sequence, a triplet sequence, like CGG, AGG, something like that. So Cas9 needs this, all Cas, you need to design the guide RNA very adjacent to that site, PAM site or PAM site, where that Cas9 can identify it. Okay, so for example, Cas9 creates a double-strand break, and that break attracts what? Exonucleases. And the exonucleases will chop off whatever is in the middle. Okay, the double-strand breaks will get attached to the exonucleases, and a part of DNA is removed from that. So there are two ways to repair. One is called non-homologous end joining. That creates large deletions. And then that would be filled with the random nucleotides that disrupts the gene function, because random nucleotides are added to that. Whereas another way is homologous directed and directed repair, homology directed repair. Or you can send a new template. This template will act as a guide to fill whatever is destroyed in the middle, whatever is chopped off in the middle. In other words, the gap that you created using CRISPR-Cas9 technology, you are filling with gene of your interest. So this is collectively is genome editing. Okay, that is question number 18. So what is personalized? What are the ethical considerations? This is easy question. You guys know. What are the ethical considerations for personalized medicine? The DNA sequence, who has to create a DNA sequence, who has to interpret the DNA sequence, and how to exchange the DNA sequence, because privacy is the most important. You cannot freely share it, right? So who can own your DNA sequence, and whether you need to sign that off. So all these things, like if you have very healthy DNA, you would like to share it with the companies to show that you have no problem, so you can get that insurance policy for very cheaper prices. You can get good deal. Whereas if you have problems that you are predisposed to get a particular cancer, you can get that cancer, by exposing yourself to a specific environment. So you would like to protect your information. You don't want to share your DNA information. So that is privacy and confidentiality. And discrimination can happen, genetic discrimination. Somebody will say that we cannot recruit you in a particular job because you are genetically not competent enough. Then, so you can easily get stressed, or you are predisposed to post-traumatic stress disorder. Then you cannot get high-end military jobs because you get stressed so easily. So there is Genetic Information Non-Discrimination Act in the United States. Probably you all know the Non-Discrimination Act for the genetic information. And also, there has to be consent for sharing the information. And there is a psychological impact. Like once we sequence your DNA and tell you that you have these issues, that will cause anxiety, fear, and distress. You are highly motivated student and you have goals. You want to become a doctor, you want to become a scientist. But if you come to know that you have a predisposition to cancer, a driver mutation, that is emotionally challenging. You don't want to know that information, you see. Equity and access is another thing. Once you have this information that you have a deletion, you are getting sick because of a rare mutation. And though it is not a completely, deleterious mutation that or lethal mutation, you may have a rare disease situation that you may feel nauseatic or you may get stressed. For that, you need personalized medicine specific to that deletion. So, if you have this information, if physicians are knowledgeable to understand your DNA mutation, then that is very useful to cure those rare diseases. So this is next question is question number 20. Explain how DNA microarray and RNA sequencing technologies are different. We discussed in the class very nicely this topic. DNA microarrays are small arrays, okay, small epoxy surfaces, a postal stamp size epoxy surfaces, where millions of fragments are spotted on it. Some kind of DNA inkjet printers will print nucleotides onto those epoxy surfaces. So what are those nucleotides? Are oligos, in the sense, short DNA nucleotides, short stretches of nucleotides, say 15 nucleotides or 8 nucleotides. So a gene can be divided into multiple fragments, and all those fragments are converted into oligos and spotted in an array, like in replications, in a lot of duplications. So the DNA from the sample control cell and then diseased cell can be isolated, RNA can be isolated, and RNA can be loaded, like one sample can be loaded red color, one sample can be loaded green color fluorescent, that is Psi3, Psi5 dyes. Psi3 gives red color, Psi5 gives green color, and both can be mixed together and then hybridized to that chip. So if it is green color spot, that means that is occurring only in the Psi3 loaded. Psi5 is for red color, sorry. Psi5 is for green color. Psi3 is for red color. So you can see looking at the spots, your high throughput software can measure the intensity of color, whether it is green or red. If a particular gene is not connected, it is both red and green binds to it, then it gives yellow color. Or more red genes are binding, lesser green are binding, that gives a different color. More green and less red are binding, that gives a different color. So based on the color intensity, you can understand the gene expression in the microarrays. So what are the advantages of microarrays? Rare genes can be identified. Because rare genes are also spotted there, you can understand them. Whereas RNA sequencing technology is isolating RNA, converting it into double-stranded RNA and a DNA, that is because using the reverse transcription methods. So for that, all messenger RNAs have poly A tails. So poly A tail specific oligo can be designed, poly DT tail. This DT tail can be used for reverse transcription of that RNA. So this double-stranded RNA DNA duplex can be used for RNA sequencing technology. That is, you can remove that RNA strand, you can synthesize, you can create a hook at the end of the DNA, first strand DNA, and synthesize the second strand, and then you can make libraries with that and sequence it. So what is the difference between the DNA microarray and RNA sequencing? DNA microarray, you have the distinct number of spots there. You cannot get new technology, new RNAs, new transcripts. Whereas RNA-seq is dynamic, that is sequencing. So lots and lots of different kinds of transcripts can be realized at the end of the sequencing experiment. Whereas rare transcripts cannot be seen in RNA-seq because we will see only very high folding expression differences, like the genes that are showing up clear differences. We would like to consider in RNA-seq data. DNA microarray data is also subjected to bias by hybridization techniques. Okay. So this question number 21 is gene cloning and the role of restriction enzymes and plasmid vectors. So we discussed that in detail, that creating of recombinant plasmids and then creating the recombinant bacteria. So there are two kinds of plasmids once you carry out the experiment. Some of the plasmids never got the foreign DNA, and some of the plasmids are cloned with the foreign DNA. Okay, so recombinant plasmids and non-recombinant plasmids both can be used to transformation inside the bacteria. Then you have three kinds of bacteria. Bacteria that do not have plasmid at all. Bacteria that do have non-recombinant plasmid, are they not useful, though they are recombinant bacteria, the plasmid is not recombinant. But we need to identify the bacteria that has recombinant plasmid. So using the antibiotics, you can kill the bacteria that do not have any plasmid, but you can use the LAC-Z, a reporter gene, in the cloning site that expresses beta-galactosidase, and in presence of X-gal that turns that particular bacteria into blue color. So the most important role of restriction enzymes is you need to cut the DNA and the plasmid with the same enzyme so that both can be pasted together. Okay. So if you want, you can draw figures also, or you can write. I do not mind. You can write few points correctly, whatever we are discussing here, or you can draw the pictures to get the points. So out of 25, you need to select only 20 of them. 5 are the choice. So what is the model organism Caenorhabditis elegans? That is a very important model to understand cell lineages and their fate. So Caenorhabditis elegans has a distinct number of cells. So the hermaphrodites are larger and males are smaller. Like some 1,000 number is there in hermaphrodites, 1,000 cells totally, and some 900 cells in the males. So each cell position is so important. So they are the, you can distribute all those cells into lineages, like A lineage to Z lineage, and each lineage has distinct function. So in the class, we discussed how the vulval tissue, vulva forms in the worms. That is Z cells, ZAA, ZPP has to interact. So you can understand this interaction in two ways. One is ablation. You can ablate a particular cell, which is in the particular position. Or you can create mutants, especially a transmembrane protein called Lin12 is very important for interaction of ZAA and ZPP. This interaction is very important for downstream signaling. Okay, so that signaling is very important for formation of vulva development. So what can happen here? You can either study the mutants or you can ablate to understand the cell lineages. That is what you are going to discuss in question number 22. The genes especially are epistatic or hypostatic. In the pathway, in the cell lineages, how the fate determination happens is, say, gene A, gene B, gene C, gene D. Gene A can be epistatic to gene B, or gene B is hypostatic to gene A, or vice versa. Gene B will be in the beginning, and that is epistatic to gene A. So to understand that epistatic relations, we need to create mutants. Okay. So the genetic mechanism of vulval development is that LIN12 is one gene, and then what happens? One of the lineage cells will be producing LIN3. LIN3 is a ligand again. That LIN12 is between the GPPPGAA, okay, and then downstream to that, a lineage produces LIN3. That LIN3 is kind of signal molecule or ligand for a receptor on the P6 cells. In the P6 cells, especially has a receptor called lead-23. So lead-23 and LIN3 has to interact to produce bunch of cell lineages that finally end up into a vulval cell. So here we discuss anchor cells and vulval cells. And in vulval development, we clearly say LIN12 and lead-23, LIN3 and lead-23, etc. So the define epistasis, how epistatic relations can be used to map genetic pathways. This is, we discussed 6 or 7 different mutations are identified, out of which how to identify which is epistatic, which is hypostatic. You need to cross 2 different mutants, and then you need to understand which is the mutant that is expressing in the double mutant. When you are crossing 2 mutants, you are creating a double mutant. In the sense it has both mutations. But what is its phenotype? Whether it is mutant kind of A or mutant kind of B. If it is mutant kind of B, that means mutant B is masking the effect of mutant A. If the A phenotype is expressing in the double mutant, A is the one that is masking the effect of B. Okay. So whatever is epistatic, that is in the beginning. Whatever is getting masked, hypostatic, that is next. So using that, we establish genetic pathways. Okay. In the book or in the PowerPoint, it is a little bit more detailed that what genes are used and how that is done. You can use that model and explain, or you can just explain clearly how epistatic and hypostatic is determined. So the last question is Drosophila melanogaster embryogenesis. That beautiful YouTube video also is there. So what happens? The egg is hatched and it has a single nucleus, and that single nucleus undergoes multiple mitotic divisions, producing some 200 nuclei, and all these 200 nuclei will go to periphery and create a blastoderm formation or syncytial development. That is the cell wall kind of invaginating into the nucleus and creating our peripheral cells, and that one further kind of multiply all those cells and produce a what is that, glastula. And then there is bicoid gene that is coming from the egg. You see, the egg, before the egg and sperm fuse and produces a zygote. In fact, we should not call it ovocyte and sperm. In the ovocyte, there are mother-specific genes. So once the sex occurs and then a zygote is formed, that zygote becomes embryo. Inside the embryo, there are mother-specific genes already there in the cytoplasm. They are called maternal genes. One of the most important maternal gene is bicoid. So that bicoid is a activation protein for several genes that are in the zygote nucleus. So the zygote has a nucleus that has both maternal and paternal genes. Okay. So those paternal and maternal genes that are there in the zygote in the diploid condition, they are silent first. So in embryogenesis, what must happen? This bicoid, which is a maternal gene that came from the oocyte, and after fertilization, the zygote is called embryo, and the embryo develops into egg, and the egg hatches. Okay. So once that egg is hatched, blastoderm develops, and blastoderm will become total worm, larvae. So in that larval development, this bicoid, which is maternal gene, will interact with the zygotic genes. That is really amazing. Then that zygotic genes will trigger to produce anterior and posterior sites of the Drosophila melanogaster larva. Even Monday's lecture, I will be dealing a little bit of this. So tomorrow's class, since the test is on Wednesday, in tomorrow's class, you need any explanation for these questions? I can go through again, whatever you want. Okay. So any questions so far? I will send you this PowerPoint, this recorded version. And also you will have tomorrow's class also. If you have any questions, you need any special preparation, you can ask me to discuss. So we can go through again some of these questions. So what do you think? So you guys need to pick your choice of questions and then start building up a synopsis. What are the points, important points we discussed? You can draw figures or you can put the points to get the point, to get full points. So any questions so far? Or you want me to go through any of the topic again? No questions. Terence, are you still there? Okay. Sounds good. See you folks tomorrow. So before tomorrow, you come with a synopsis. Okay. Or you pick the questions out of 25, which of the 20 you are going to answer. And then start writing whatever you can in that, some points. And then if you want, you can meet me tomorrow anytime. Tomorrow afternoon, I'm available in my office. I will look into your synopsis and help you to add or to focus some points clearly, or we can discuss in the classroom also. And I will be sending this recorded version. Okay. All right. Have rest of nice weekend. I'm closing down. If there are no questions. Hello, Rishi. How are you feeling?
I'm fine, sir. How are you, sir?
Good, good. Happy Sri Ram Navami.
Oh, yes, sir. Happy Navami.
How is your pain? Is better
Uh,
now?
yes, sir. It is better.
Okay, good. All right, then. See you tomorrow.
Yes, sir. Thank you, sir. Have a nice day.
You too. Have a great weekend.